So welcome everybody. I'm Marco Dalmaschio from the University of Padova, and I'm introducing you the webinar of today. You have to know that this FNIP webinar are a bottom-up initiative that is somehow merging a different field of research. So this is a this kind of research in neuroscience is somehow getting at the convergence between photonics, microscopy, and data analysis. And the purpose of this kind of webinars is to bring together established neuroscientists, research in the field of the biophotonics and also material engineering to basically state what the current state of the art of the technique and the research and what this different field can contribute on the research in the future. So when I say that this is a bottom-up initiative, I basically refer to the fact that this is an initiative that has been promoted from a group of younger researchers from different departments here in Padua and from different institutes. We have the Department of Biomedical Science of the university. We have the Institute of Neuroscience from the CNR, the National Research Council in Italy, and we have a researcher from the Department of Physics and Astronomy. And what is very cool, and the work of this team has somehow involved a very large network of scientists across the world. And when I say a very large network, I refer to also people that have been contributing to design the panel of speaker or presenter that have been selected in the last event. So I hope this initiative can somehow keep going because it's very interesting to see different kind of fields that are somehow meeting together in this kind of webinar. So when I mean that the network is rather large, or the speaker is really involving research in the United States, is involving research in Europe, and is always involving research in Saudi Arabia. So we are with a very open perspective. And if you look at the website, the program is really covering different aspects that are somehow instrumental for the research that we are interested in. So I don't want to take too much time to the speaker, but what I want definitely do is to really thank the speaker that have been joining us today. So it's very important for us to establish this kind of network, and I'm really happy that they can manage to join today and be involved in this kind of initiative. So you should be aware that this webinar are recorded, but more importantly, that this webinar are available on the web page of the initiative. So if you look at this link, basically you can find all the program or the schedule for the webinars, the one that are have been already done, and the one that are going to be in the next months. But especially you can find the contribution from the speaker. So I really invite you to have a look. Without taking further time, I want to pass the stage to the colleague that I introduced in the speaker. I want to thank everybody for joining. I want to thank the speaker for being here today. And we look forward for your presentation. Thank you.
Marco, could you please stop sharing the screen?
Sure.
Thank you. Okay, so hi everyone. Today the first speaker of this webinar is Sebastiano Curreli. A couple of words to introduce him. So he got his master's degree in medical and pharmaceutical biotechnology at the University of Genova in 2011. Then in 2015, he obtained his doctoral degree in neuroscience and brain technology from the University of Genova, working at the Italian Institute of Technology under the supervision of Alexander Dityatev, working on the optimization of strategies for directing reprogramming of glial cells to neurons. Then he joined Professor Dityatev at the German Center for Neurodegenerative Disease in Magdeburg, Germany. Then from 2016, he's a member of the Optical Approaches to Brain Function, led by Tommaso Fellin at the Italian Institute of Technology, where he investigates the basic processes underlying information encoding in the brain, focusing on the role of non-neural circuit elements in parallel information encoding. So, Seba, it's a pleasure to have you here, and please, the virtual stage is yours.
You have to enable your, thank you.
Done. So, good afternoon, everyone, and thank you, Marco, for the kind invitation. It is a pleasure to spend time discussing about the recent research I managed to achieve in Tommaso's lab. And in this presentation, I'm going to discuss with you about information encoding processes in the brain. But as Marco said, I will do it under a different light, focusing on non-neural cells and specifically on astrocytes. So to successfully interact with the world, animals rely on encoding physical features of the environment and use this information to plan and perform complex behavioral decisions and actions. Understanding how the brain handles information streams of physical features from the external world to inform computations that guide behavior is one of the outstanding questions in the modern neuroscience, systems neuroscience. Starting from this extract from the work of Perkel and Bullock, we can see that the notion of information processing brain is with us since a while. As a rough approximation, we can envision as if the brain was a machine hosting a set of processes that underpin how inputs entering the brain through perception are exploited to inform output actions of the organism. At its core, the notion of information processing brain relies on the assumption that neurons map external stimuli as sequences of stereotyped electrical action potentials. And the statistical properties of action potentials are then the putative building blocks of the neural code. Here, you can see one of the most famous examples of neural code. During spatial navigation, some cells of the hippocampal formation discharge spikes at specific spatial locations. These spatially clustered spikes are then the hallmark of the firing patterns of place cells. These cells have been classically identified by heuristic descriptions of their average firing field shapes, leading to the proposed model that these cells provide a cognitive map that constitutes an internal representation of space. Similarly, a plethora of other navigational related cell types has been described, including head direction cells and grid cells. And these findings constitute a fascinating and elegant body of work that has been awarded the Nobel Prize for Physiology and Medicine in 2014. However, maps and coordinate systems are semantic categories that we use to describe by analogy patterns of our observations. Yet, often we don't have an a priori model to guide our understanding of a phenomena. And for this reason, to reverse engineer a complex system such as the brain, it would be advantageous to have a general framework to quantitatively describe relationships between stimuli and brain responses. A powerful tool we can borrow from the field of mathematics is information theory, pioneered by Claude Shannon. Information theory relies on probability theory and explicitly deals with measures of information and their application in the study of communication systems. Suppose that we were to send a message a stochastic variable x. If x has only one value, there is only one possible message, then no information is transmitted by sending that specific message. Thus, it is intuitive that the amount of information that we can obtain from a message is linked to its uncertainty. And we can define a function, entropy, that measures the amount of uncertainty inherent to a random variable as a function of its probability distribution. If we were to send the values of x along a noisy communication channel to a receiver y, we could describe probabilistically the transition probability of receiving the message y1 when sending x1 as a message. Observing the message y reduces our uncertainty about the source message x, and we can quantify this as a conditional entropy of x given y, which describes the residual uncertainty of the message while we know the readout of y. Knowing the intrinsic variability of our message, which is entropy, and the conditional entropy of our information transmission process, we can quantify the average reduction in uncertainty of the source message due to the knowledge of Y. And this quantity is known as mutual information or Shannon information. In neuroscience, we can use these powerful tools to approach the neural coding problem by analogy. And for consistency, I will stick to the information encoding or to the spatial information encoding paradigm that I described before. We can define the stimulus entropy as a quantitative description of the information content in the stimulus itself. For instance, if the amount of uncertainty of the stimulus is small, the entropy will be small itself, while, for instance, if the amount of variability of the stimulus itself is big, the entropy function will be high. For instance, if we can envision this as the exploration patterns experienced by animals, and a distribution of exploration patterns that is covering the whole arena would yield a high entropy. Conversely, if the mouse would stick to a corner of this arena, we would have a small entropy value because the uncertainty is quite small itself. At this point, we can look at neural responses. And for instance, these blue dots that I just modeled over here are scattered throughout the arena regardless of position. And we could use these responses to quantify whether their observation would convey some information about the stimulus itself. This is called stimulus equivocation, which is the conditional entropy of the stimulus given a neural response R. Computing mutual information, we can then quantify the reduction of uncertainty about position provided by this set of responses. I would not be surprised if we were to quantify this set of responses, for instance, to obtain a very small amount of information in comparison to the actual entropy value of the stimulus itself. Let's look now instead of this to these red dots that indicate that simulate a neural response similar to the one of a place cell. It is trivial to see that on average, our uncertainty about the stimulus is much smaller when observing these responses, because when we see these spikes, it is quite likely that the animal is passing by this area of the arena, leading to a reduction of our uncertainty about the stimulus. And therefore, it is trivial to infer that the information encoded by response 2 is much bigger than the response encoded by R1. This very simple example highlights the power of information theory when applied to neuroscience, because it allows to describe a quantitative stimulus-response relationship without the need of an a priori model for the inner working of the circuitry or the encoding itself. Information theoretical measures are often complemented by decoding approaches, where we can define decoding as a prediction of a stimulus or a behavior that a stimulus or a behavior elicit in a particular neuronal response in a single trial. In this context, decoding models are built on the knowledge of the population of neural responses, and we can map stimuli on the n-dimensional space described by the n neural responses observed. The decoding algorithm is then optimized using a subset of the data to predict which stimulus elicited the population response provided as input on a single trial. And the remainder of the data is then used to test the predictive capabilities of the model. Results of decoding analysis are often summarized in confusion matrices. Here are the presented stimulus lies on the x-axis, while the decoded stimulus lies on the y-axis. And each cell of this matrix is color-coded as a function of the percentage of occurrences classified as that cell. As you can see in this example, a faithful decoding would yield brighter diagonal, indicating that we could infer something from the stimulus, about the stimulus from the decoded information. However, if we were to stick only on to the data that lies on the diagonal, we would be receiving an incomplete description of the decoding performances, because we would not have information about what's lying close by to the diagonal itself. And to overcome this limitation, we can rely again on mutual information to quantify the information about the stimulus that we gain observing the decoding predictions. With this set of tools, I decided to join the dark side of neuroscience, investigating information encoding beyond the neural circuits, because indeed, brain complexity extends way beyond neurons. And as beautifully described by Ramon and Cajal, astrocytes and glial cells in general have a very deep anatomical relationship with neurons. And he even postulated some functional assumptions about that. And it was more than a century ago. And indeed, astrocytes are the most abundant non-neuronal cell type. They are non-electrically excitable, but they exhibit a form of activity as fluctuations of their intracellular calcium concentration. As you will see from the talks of Natalie and Alfonso, calcium signals of astrocytes regulate a plethora of functions, including neurovascular coupling, synaptic plasticity, and network dynamics. In this cartoon, you can see that calcium signals can be quite heterogeneous. They can be restricted to subcellular regions of the astrocyte, as the soma or the processes, or they can involve the whole cell. Using functional calcium imaging, we can observe the temporal profile of astrocytic calcium dynamics at specific subcellular regions. Importantly, calcium dynamics relate to neuronal activation, neurotransmitter release, and correlate to neuronal responses in vivo. For example, in classic experiments in the somatosensory cortex, astrocytes respond to whisker deflections with calcium increase, suggesting that astrocytic calcium dynamics can be involved in information encoding, at least in primary sensory areas. With this in mind, we formulated the hypothesis that astrocytes may encode navigational information in the calcium dynamics. And to test this hypothesis, I coupled virtual realities, a virtual reality navigation paradigm with two-photon calcium imaging, where mice run along a virtual corridor provided with visual features. And at the end of each run, they get teleported back at the beginning of the track. To visualize astrocytic calcium dynamics, we implanted a chronic window above the hippocampal CA1 area, and we specifically labeled astrocytes with GCAM6F. This enabled us to record astrocytic calcium dynamics with subcellular resolution, allowing to extract calcium activity at specific subcellular regions while knowing the mouse kinematics parameters. Then we were in the position of tackling this problem using information theory approaches, as I described before. And we computed mutual information for the signals we recorded from astrocytic ROIs. And we evaluated whether these signals are reducing our uncertainty about the mouse position. We found that approximately 44% of astrocytic ROIs significantly encode information about position. These black lines over here are the average fluorescence signal as a function of position for a sample of 5 astrocytic ROIs encoding spatial information. And I refer to this black function throughout the talk as the response profile. In gray, you can see the preferred spatial region of response that I will call the response field. At a population level, we can represent each ROI as a row and color code its response profile. And we can order rows according to the center of the response field, here marked as a yellow dot, while the spread of the response field is marked with purple dots. I've tested, of course, how robust these relationships are using several resampling approaches. Here, for instance, I've arbitrarily divided these trials in half, being either even or odd, and we can see that the overall pattern is quite compatible, demonstrating the robustness of the response profiles. On the right, you can see quantifications of response profile features, such as the median width of and the distribution of response fields. And importantly, we can highlight that the difference in response field center between each decimation approach and the actual dataset is centered in 0, quantifying that actually these responses are quite robust. As reported in the literature, astrocytic calcium signals have been shown to be organized at the subcellular level. And for this reason, we moved to investigate the subcellular organization of astrocytic spatial information encoding. We started in this example ROIs of an experimental session are coded as a function of their response field position. And as you can see, there is quite diverse scenarios, with some astrocytes showing multiple ROIs with the same preferred position, while others showing quite diverse scenario. We tackle this quantitatively, and we first focus on measuring this relationship within single astrocytes as a function of anatomical distance over ROIs. And we found that the response field center distance grows as a function of the distance from the sum, indicating that within the same astrocyte, there can indeed be some differential spatial tuning. Then we quantified this the topological organization at the population level, measuring pairwise features. And here we have the pairwise distance of response field center as a function of the pairwise distance of the ROIs. And we can see that this difference increases within the 40 micron regimen, then approaches a plateau, which is approximately 40 centimeters throughout the anatomical distances we analyzed. Complementary temporal correlation between ROIs decreases within similar dynamics. And overall, this data indicates that individual astrocytes can encode multiple spatial fields across different subcellular compartments. Then we asked whether the information contained in the astrocytic calcium signals could be used to decode the information about the mouse position. And we formalized this question as a classification task. We used population signals of astrocytic ROIs coming from an experimental session to train a support vector machine, asked to classify the animal's position in discrete regions of the linear track. We changed the number of discrete positions that the decoder had to learn, and we refer to this parameter as spatial granularity. Classification results can be summarized in diffusion matrix, as I mentioned before, and in the x-axis we have the actual position, and in the y-axis the predicted one. As you can see, when the decoder is asked to learn to distinguish between 4 positions, the performances are quite high, and we can see that performances are quite reproducible across all granularity tested. Quantification of decoding performances has been done, as I was mentioning, with information theoretic measures, and we consistently observe above chance decoding, meaning that we are able to infer the animal position from the population activity of astrocytic ROIs. Importantly, errors of the decoder are centered close by to the diagonal, meaning which is a positive, a bona fide control of decoding. Importantly, if we were to disrupt correlations between ROIs, we could find, we would find a significant decrease in the amount of information encoding in the population vector, suggesting that correlations between ROIs as a relevant source of information within the astrocytic network. At this point, we asked, how does astrocytic spatial information encoding relate to the neuronal representation of space provided by place cells? And to tackle these questions, we upgraded our preparate to perform simultaneous calcium imaging of astrocytes and neurons during virtual navigation, labeling astrocytes with GCAM6F and neurons with JRCAM1A. This enabled us to visualize simultaneously spatially modulated astrocytes and place cells that here you can see again color-coded as a function of their place field position. Thus, we are now in the position of tackle the question which I just posed. So to investigate the relationship between these two coexisting spatial representations. At a population level, both astrocytic and neuronal response profiles show a spatial pattern along the linear track. And we see that neuronal place fields are sharper than astrocytic response fields, and that they have a different distribution along the linear track. At this point, we asked whether neuronal and astrocytic spatial representations relate to their anatomical layout. And we examined the relationship among pairs of ROIs made by a neuron and an astrocyte as a function of their anatomical distance. We found that astrocytic and neuronal signals show small but significantly positive temporal correlation, which was approximately constant regardless of pairwise distances. Similarly, the differences between their response, the preferred response position, the response field center, was largely independent by the anatomical layout. And these findings thus suggest that neurons and astrocytes might have distinct spatial tuning. With this in mind, we tested whether neuronal and astrocytic spatial representations could be the substrate of population coding. And again, we relied on the formalism of information theory for a quantitative approach. We studied information we studied the mutual information about stimulus given a set of responses. And here I will focus on set of responses made of pairs. First, we found that on average, observing the response of two ROIs provided more information about the stimulus than the sum of the independently encoded information. And this was true regardless whether pairs were composed by exclusively astrocytic ROIs, neuronal ROIs, or by one ROI of each type. Importantly, we found that a significant fraction of pairs suffered information loss when disrupting correlation through trial shuffling. With this in mind, we used an information breakdown approach to characterize the source of gaining mutual information, which can be thought as the difference as a differential term remaining after accounting for the sum of the information provided by each single element of the response array. Delta I can be decomposed to understand whether its value is dependent independent from the stimulus, in this case position. And we can see that in pairs encoding by correlation, the delta I term is mostly related to the stimulus itself, so this blue bar over here, okay? And consistently, we found that correlation magnitude between pairs of responses was modulated inside and outside the respective response fields. Overall, this data suggests that astrocytes and neurons encode complementary information about space, relying both on the diversity of position tuning and on position-dependent modulation of their correlations. This set of results prompted us to test whether using both neuronal and astrocytic population signals, we could improve the performances in decoding of mouse position. And again, we formalized the question as a classification task, and we used support vector machines that were either provided with neuronal or astrocytic signals. As before, we performed the classification as a function of granularity, and the decoder was fed in turn with astrocytic responses, neuronal responses, or both. Results are summarized in confusion matrices, and measuring decoded information would allow us to consistently to what we observed with pair analysis. We found that removing either source of information resulted in a significant drop of the decoded information across all granularities. Moreover, in line with the information breakdown, the disruption of correlation in the population vector consistently caused a significant reduction in decoding performances, indicating that the modulation of correlations across these two population vectors acts as a source of information. And overall, this finding indicate that astrocytes provided complementary information about the stimulus that is not present in neuronal dynamics. Thus, to summarize and conclude, I showed you that astrocytic calcium activity is reliably modulated by the animal's position. Thus, spatial information encoding do extend beyond neural circuits to neuronal networks. Somatin processes participate in spatial information encoding, showing distinct spatial tuning. Astrocytic calcium signals provide significant information to decode the mouse position. And during virtual navigation, CO1 neurons and astrocyte encode spatial information at a population level that is complementary. Thus, this result demonstrates that these two subset of cells are the subset of a population coding. However, it is crucial to be aware of the limits of these findings, especially because correlation do not mean causation, okay? And for this reason, we're keeping the foot on the gas to address the following open question. Namely, what are the network effects and the behavioral effects of acute perturbation of astrocytic signals on mouse behavior? We are further extending these observations in bidimensional, more naturalistic spatial navigation. And I'm actually in the process of implementing mini two-photon microscopy technology developed in the Moser lab. And last, it would be very relevant to extend these findings to other forms of information encoding in the brain, because these principles might not be related only to the hippocampal network. So far it has been a very fun journey that it would have not been possible without my advisor, Tommaso Fellini. And I have to give a huge mention to Jacopo Bonato and Stefano Panzeri from the Neural Coding Lab, which provided great analytical tools and insights throughout this project. And importantly, I want to mention Sara Romanzi and Pedro Lagomarsino and Luca Sita from the Optical Approaches to Brain Function for their invaluable contribution throughout the project. And of course, all of you for your attention.
Thank you, Seba. Nice talk. So we have time for a couple of questions, perhaps from the audience or from the guests, perhaps, or otherwise I would go myself with a couple of questions. Yes, Alfonso, please.
Great talk, Sebastiano. I have a question.